All right, welcome. I hope people can hear me also in the back. It's my big pleasure to welcome everybody. This officially is Nanion's the biggest ever Nanion user meeting, I have to say. And I'm really relieved we earlier in the year decided to not do it on the third floor in our premises like we normally do, but to go here to a larger conference room to fit everyone in. Right, no coincidence, it's a big user meeting because it also was a very big year for ion channels in 25. We started out in January with the FDA approval of the sodium 1.8 inhibitor called Jonavax by Vertex, which is the first non-opioid pain relief drug in more than 2 decades. I mean, NME17 has been screened up and down for a long time, nothing to show. So this is a great breakthrough success for ion channel drugs, and clearly made a lot of press. And around that, there was to see a lot of investment in sodium 1.8. Companies like Latigo Bio and Side One have received large funding rounds for their sodium 1.8 programs. So a lot of activity in that space. And a little later in the year, there was a second very big news in that Eli Lilly did acquire Side One for $1 billion, in particular for their 1.8 program. So that was a direct response to the FDA approval of Jonavax, and clearly sort of a validation of where the next generation of pain relief drugs, non-opioid pain relief, will go. So that was all the craze in the beginning of the year. But actually, in between these two big events, there was yet another in that the FDA approved a further ion channel active drug from Soleno Therapeutics, a KATP channel opener that is used in Prader-Willi syndrome. That's a rare genetic disease disorder causing uncontrollable hunger and obesity. So that was the second ion channel drug to be approved. And then in June, there was a third, this time a TRPM8 agonist for dry eye disease called Triptyr by Alcon. So actually 3 drugs in 1 year. That is a lot, actually in between January to June. So that to me is kind of a, almost an historic turning point for ion channels. Really great success. At least I would like to believe this is a new big wave in ion channels. And in that regard, I would also like to cite a well-known ion channel expert who has probably similar opinions there, in saying that in the ion channel field we haven't seen this level of activity in years, and this validates the field's transition from difficult targets to preferred targets. Another thing I'd like to believe is true, and I certainly can see this. There's good reason why that it should be true. We've seen a lot of activity and investment around ion channel companies, buyouts, the Side One deal being just one of them. There's plenty, and there's plenty more than listed here on the slide. We've also seen in this year a lot of new ion channel companies starting out and being financed quite well. Also, that is a much higher activity than we normally see in the ion channel field. There's plenty of new companies. Want to mention maybe Drake Therapeutics from Wales, from the UK, starting off the ground with $140 million. That's a big round for European biotech. So really great to see all the success around ion channels. And I think it's fair to claim that ion channels as a drug class are really having a moment here. And seriously, I think this is just the beginning. So there's going to be more, and I still like to believe so. If you are interested to stay in the know around ion channel drug discovery and clinical development, I would really recommend you read the quarterly reviews of Mark Rogers, technologists that are published, on LinkedIn and elsewhere. By the way, Mark also is in the audience, so a good chance to chat him up. Hello. I never say a word again about Bloomberg. Don't you worry. Right, but this is really, to keep up to date, a really great resource. So other than that, I mean, some of you probably know, I will not get tired talking about organella electrophysiology and in particular lysosomal ion channels. They were also big this year. There's strong interest both from academia but also from drug development. And one exciting news in that regard was the launch of Tenvi Therapeutics. They launched in January with a 200 million investment to develop drugs for TRPM1 and TMM175. So yet another ion channel company. Clearly hot targets. And if you are similarly excited about lysosomal ion channels and transporters as we are, I would like to invite you to take a look at a recent review that we published in Bioelectricity together with a lot of the key scientists in the field, going into much detail, really good resource for things. I will not go any deeper in that regard, but our next speaker, I'm very pleased to have Christian Grimm here from University of Munich and also Oxford University. He will talk about high-throughput organella EFIS on lysosomes, so there will be a lot more detail in your talk, Christian. But as said, lysosomes are clearly hot, but it's not only lysosomes, it's actually all the different organella structures that are kind of big interest, and I think this is where things are headed. In particular, ER or mitochondria are in large demand. These are some preparations from a company that we've been working with for more than 2 years, Oriya Bioscience. They specialize in making preparations of organella membrane for drug discovery and research. And these are beautiful ER membranes here ready to be patched. Really cool stuff. Definitely an interesting space to watch. And also we have received quite recently some mitochondrial preparations from Oriya. And here on the left, you can see we throw those on the synco patch. And these actually patch quite nicely. Very high success rate recordings. This is early days, but we are quite excited about that. And another partner, Axam, a CRO from Italy, also is working on mito patch. And on the right, you see their preparation. They make them themselves and also throw them on their synchro patch, or that is to say on their 2 synchro patches, and make some nice MCU current recordings. So clearly rather exciting field. The whole organella and mitochondria is one of the key things there. Also, would like to mention that also in the audience today is Alexandre, the CEO of Oriya Bioscience, as well as Jeff, Jean-François Roland from Axam. So if you're interested in organella EFIS, these are also good people to talk to. Just to mention that this is how they look like if you don't know them. Right. Then of course, well, AI is everywhere, also in ion channels, not surprisingly. AI is used to predict structures and to come up with the de novo designer pores or de novo channels. There was recently a very nice work on a de novo designed voltage-gated ion channel, very impressive. And of course, it's also used in accelerating drug discovery, de novo designed ion channel modulators. And that's something we will hear from Stefan Pless. I'm also very pleased he's joining us today. And Stefan will tell us about how he uses deep learning tools and high throughput electrophysiology to design ion channel modulators. Right, there's a couple of more talks using HDS APC. One from Ann-Katrine from University of Wollongong, down under. She will tell us about chloride transport in circulating tumor cells, as well as from David from University of Zurich, exploring heterogeneous cell populations using automated patch clamp. Right. I'd like to share a couple of news from our high-throughput instrument, the SynchroPatch. Some of you may be familiar with the work we did on mechanosensitive assays. We developed a very nice high-throughput assay using the SynchroPatch pipetting system to stimulate piezo1 for mechanosensitivity assays. And well, I'm quite pleased to share or to brag about, to be honest, that we placed an instrument at the kind of epicenter of the piezo world in San Diego at the Scripps Institute in Adem Pataputian's lab, who is here happily receiving his new toy. He apparently is quite excited about that, and we certainly are as well. So we're looking forward to see getting results from Adem's lab on piezo using the SynchroPatch. Another news from the SynchroPatch I would like to share is regarding our optogenetic stimulation capabilities for the SynchroPatch. We have this year launched or released a new version of this, coming with full-blown 384 activation sites. You can run the full plate with 384 LEDs. So you see the 4 cylinders here, having 96 LEDs each. You can choose from a wide range of different wavelengths, and you can actually switch in between 2 wavelengths per well. So you have a dual wavelength operation, which I'm sure will be received quite nicely in the field. This tool is already used by a number of our customers, and we've seen some very nice papers coming out this year using the optogenetic stimulation, mostly working on channel rhodopsines and characterizing them, well, using those to make patient restoration using optogenetic control. And besides channel rhodopsines, this tool is also used for work on photoactivatable compounds and photoactivatable toxins. Quite interesting, also nice papers from Michel Dewart's group. And I'm not going to tell you any more on that because Michel is presenting on precisely this optical control of ion channels using light to stimulate. And yes, we are quite excited about the light stimulation. So we've chosen to make that our theme for this year's Nanion user meeting, beer mug, a stein carrying this nice channel rhodopsine. So you will have a chance to use that later today. And of course, also take one with you if you choose to do so. Some of you have already tried the mug last night. And hope you had a good time with that. Right, talking about light all the time, I would also like to mention a new little tool we have for the Orbit Mini, our bilayer platform. It's a fluorescence kit that enables the combined electrical and optical recording from lipid bilayers using the bilayer chips on a microscope stage. And people hear what are great applications you can do with this kind of combined setup. From Jan Behrens from Freiburg and Mariana from EPFL in their talks, even in the high resolution version of these combined recordings. Generally, we've seen a ton of great things from our Orbit bilayer systems, a ton of nice work and great papers coming out. Just to mention a few of those, there was an Harpreet Singh's group. He used the orbits to really characterize and actually discover the first functional ion channels in extracellular vesicles or exosomes. Quite impressive work. Then the orbits have been used to reveal that delta-type glutamate receptors are actually ligand-gated ion channels, which was great work done by Ed Twomey and colleagues. And last not least, David Baker uses the orbits in his lab to analyze and characterize his designer nanopores and from scratch designed beta barrels. Really cool stuff. And nanopores is a big thing, of course. Well, this also is AI driven de novo design. We will hear a little more on nanopores from Anna Sua from University of Fribourg, single molecule detection in neurodegenerative disease. And yeah, with that, I would like to move on to yet another part that we do at Nanion. We are all in for electrophysiology, but that is not only ion channels, that is also transporters. And we are quite pleased to see how our transporter assay technology, the SURFER, solid supported membrane electrophysiology, is being taken up in industry and also in academic research. Over the last 2 years, we have seen really adoption in industry and CROs as well, offering services for transporter assays using the SURFER technology, which is very pleasing. The number of publications has been growing and is sustaining at a high level. Also this year, we've seen a large number of very nice publications using the technique to study transporters. And we have a dedicated transporter session at our user meeting this year, focusing only on the SSM technology. And I'm really pleased to have with us Francesco Tardini from Italy, who is one of the very, very early users of the SURFER technology. Actually, I would say kind of a co-developer of the technique. So it's great to meet him and have him present on P-type ATPases tomorrow. That will be great. We have a more recent user of the technique, that is Ines from Aarhus. She will tell us about pinnate plant transporters. And also from the industry perspective, we have Felix from Sanofi show us how Sanofi does SLC inhibition assays using the SURFER technology. And there's one last paper I would like to highlight. This year, Ron Clark, one of the elderly statesmen in the transporter field, published a very, very nice review, a historic review of the whereabouts, how the SURFER was developed, how it came to life, what kind of shapes and forms it had over the years, and also explaining very well how the technology actually works. So if you're interested in solid supported membranes or the SURFER, this is something I would recommend to take a look at. It's really a nice review, or more a love story with the SURFER, to be fair. So highly recommended. Last not least, I'd like to mention our cell analytics platforms. So the CardioExcite, FlexSite, and the Atlas system that are used for drug safety, cardiac safety assessment, contractility, and general life cell analytics. This year, we also have a dedicated session for these systems, and we will see how iPSC-derived cardiomyocytes are used in drug discovery by Terence Hebert from McGill University, and also from Bettina from Innovitro using the CardioExcite. And two quite interesting applications of the impedance spectroscopy. Julia from University of Regensburg will show us how they use that for GPCR signaling. And the last talk tomorrow will be by Tobias Enslen from Freiburg. Quite cool stuff on pore-forming toxins and looking at the life cell activity using impedance. So that will be a lot of really cool talks. And my time is up, or maybe more than up, so I'm coming to the end now. One announcement I wanted to make in terms of meetings. Some of you may know, since a few years, we're making this membrane physiology symposium in Napa Valley. We've done that every year now. That also is growing. It's a very good meeting. I can only recommend it. This year, we have also expanded that into MPS Japan and MPS China, which were very successful and very nice. And I wanted to announce that next year's meeting, MPS and Napa, will be in mid-April. So that will be a good meeting. We already have a quite impressive list of great scientists to present, including Adem Hataputian, Yamuna Krishnan, Joe Wu, and others. So just like to invite you to join if you have the time and interest. Certainly is worthwhile. But of course, now, today, and tomorrow, we are here in Munich. And this will be a great user meeting. And yeah, as I just went through the list of speakers, we have a great lineup here, and I'm really looking forward to that. And without further ado, we will now have the talks and some posters, the chance to see them instruments in the lab, and then have a nice dinner and a chill beverage together. Hope that sounds good to you, and thanks for tuning in. Let's have a great time together. Thank you.